Hi, everyone, and thanks for joining me for episode 711 of People Behind the Science. I'm your host, Dr. Marie McNeely, and in this episode, I am looking forward to resharing our conversation with Dr. Blake Meyers, initially published on April 4th of 2016. Listeners, Blake studies plant genomes and small RNAs in plants. His lab is working to better understand how RNAs are used to regulate plant cells, particularly related to plant reproduction. And in our conversation, Blake shares some great stories from his life and his research. So get ready for this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of People Behind the Science. I am thrilled to be here today with Dr. Blake Meyers. So, Blake, welcome to our show. How are you?
I'm good. Thank you.
Wonderful. Well, we are excited to chat with you today and learn more about your career and your science. But first, I want to give our listeners just a little bit of background information here. So, listeners, Blake is a member and principal investigator at the Danforth Plant Science Center. He's also a professor in the Division of Plant Sciences at the University of Missouri. Blake received a bachelor's in biology from the University of Chicago, and he went on to receive his master's and his PhD in genetics from the University of California, Davis. Afterwards, Blake completed postdoctoral fellowships at Dupont Genomics and at the University of California, Davis. And before accepting his current positions, he was the Edward F. and Elizabeth Goodman Rosenberg Professor and Chair of the Department of Plant and Soil Sciences at the University of Delaware. And listeners, Blake is here with us today to talk about his journey through life and science. So today, Blake, we are excited to get to know you, both as a scientist, but also as a person. So can you start off this interview by telling us what you like to do when you're not very busy doing science?
Certainly. I really enjoy traveling, and plant science is really international. So it means that my work is a great way to see the world. Agriculture is important everywhere. Last fall, for example, I went hiking on a wild and abandoned section of the Great Wall of China with a colleague. On a separate trip, I went to botanical gardens in Sydney and Melbourne, and over the years, I've been to many countries on all the continents except Antarctic. When I was growing up, I dreamed of traveling, and I didn't really go into science for the travel, so I was really fortunate to land in a career that has travel as a secondary benefit. I also enjoy working on my home and reading and used to cook a lot, but science is a pretty all-encompassing career, so trying to balance it together with family, the rest of life. Maybe this is more a question about what I'd like to be doing as a hobby rather than what I actually do.
So you mentioned you like to cook. Is there a particular cuisine that you're an expert in?
No, I try to avoid Italian food, though, because my wife is Italian and inevitably I don't make it right.
Right. Can never measure up to that skill, I'm sure. Gotcha. Well, fantastic to hear a little bit about your life outside of science. And I know travel is a big part of a career in science and one of the things that a lot of people look forward to. So we'll chat a little bit later about some of these travel experiences. But first, I want to dive into your science. Can you tell our listeners just sort of a high-level view of how you describe your science to someone who's not familiar with it?
Well, I guess the detail is that we work on small RNAs, but as a kind of analogy, I like to use a car because most people have a basic familiarity with how cars function, their complexity, and some of the standard components of a car. So imagine that you're living in like 1885 and someone from 130 years in the future today delivers to you a really high-end modern automobile. And your job is to figure out how it works down to every little bolt, wire, switch, dial, fuse, really why it functions in the way it does.
I think I'd be in trouble, but...
Well, as molecular biologists, we're kind of in the same position. So studying the inner workings of a cell, which is like a complex machine, maybe even more complex than a car. And while we have really sophisticated modern tools, cells are really small, really complex. So in some respects, our modern approaches seem kind of primitive. But to go with the car analogy, in order for it to function well, there are lots of controls, regulators of things like gas, brakes, activity of the pumps, volume of the music, airflow, lights, all kinds of things. So regulation of those components is really critical to the function of the car. Too much gas and you go too fast or stall the engine, too much music, you get distracted. And so cars have a lot of computers and microprocessors in them, and those computer chips send out data, process information, direct components to do things, lots of instructions. So how does this relate to what we do? Well, in our case, we're studying RNAs, and RNAs are like instructions in a cell. So RNAs send information around and control different things. And we study microRNAs, which are essentially like switches or rheostats that turn those instructions on or off or dial them up or down. If you remove those switches, the wrong instructions go out, parts of the car go haywire. And some instructions, like turning on the headlights at night, are really important. So if you screw up those instructions, the car can break or worse things can happen. Other instructions, like dialing down the seat warmers, are maybe less important. But going back to the start of the analogy, if it's 1885 and you know nothing about how a car is put together, there's still a lot of work to figure out that the switch that turns on the butt warmer is less important than the one that turns on the headlights. But you got to work them all out. And once you know, you can sort of figure out what are the most important parts. Mm-hmm. So really, sometimes small parts have major effects. If you look at the success of like Car Talk, the NPR show, the Magliozzi brothers were really successful because they had this comprehensive understanding of how all the parts of a car work together. And they could use that expertise to diagnose the cause of some really esoteric problems. Unfortunately, with molecular biology, we don't yet have such expertise because we don't really understand how all of the bits of a cell function. But that's kind of the aim of what we're doing. So in my lab, we're piecing together how a cell functions and responds, but with a focus on some of the smallest little bits, how the instructions or RNAs are regulated. And in the coming years, with the right experiments, we can hopefully put together sort of a handbook of how all the cellular parts and genes work. And maybe we can even have a radio show too.
Very cool. Well, Blake, I've never heard this car analogy to describe cellular function, but I think it's fantastic. And I think it's a really good way to kind of think about all the complex things that are going on in a cell. So fantastic analogy. I must say, you cannot underestimate the importance of that seat warmer in the winter, though. So, well, wonderful to hear more about your work. And I know as we were talking about your life outside of science, you kind of hinted at a challenge of balancing it all. And I think this is a problem that a lot of scientists face. And the work-life balance is something that a lot of students are concerned about. And it's oftentimes an overwhelming amount of stuff to get done in a day. And I think for me, motivation is key. If I'm not motivated to go into work every day and just tackle that huge to-do list, it's really challenging. So I have these little motivational quotes and sayings kind of tacked up everywhere. I'm curious, do you have a favorite one yourself?
Well, honestly, I'm not really a motivational quote kind of guy, so I don't have those. But we have a few sayings around my household that come up on a regular basis, commonly like, you snooze, you lose, which is kind of in the same bucket of expressions like seize the day. Mm-hmm. So these have a certain relevance in science because it's a really competitive field. Oftentimes you're competing to answer a big question or get a paper out or get a grant or a fellowship funded. So these aren't really motivational expressions. It's just more of a quote to explain why something didn't happen in the household or why we didn't take the dog out or missed an opportunity. But maybe a little bit more motivational is the adage from Thomas Edison that genius is 1% inspiration and 99% perspiration. So I never really thought of myself as particularly clever, particularly when I look around at the work that the top scientists in my field are doing. But I've been successful, and this is largely due to hard work by me and my lab members in particular. We're all working hard at succeeding every day. And so it takes a balance of that perspiration, but also that 1% inspiration. You've got to, I think, as a leader of a lab, create an environment that facilitates the inspiration and facilitates the perspiration.
Certainly. And speaking of inspiration, I'm curious to hear more about these people who have inspired you across your journey as a scientist. I know when I look back on my own journey, there are so many people who helped me get here. So can you share with us some of your role models or the people that you look up to?
Sure. There are quite a few. And I think everyone who succeeds can point to a mentor that made a difference. And by the time you made it further along your career, you typically had a lot of mentors as an undergraduate, as a graduate student, as a postdoc, as a junior investigator, assistant professor, and so on and so forth. And since science often requires moving at each of these stages, you end up accumulating a series of mentors. All of them are important, and they teach you different lessons about how to succeed and how to mentor your own lab members once you get to the level of running a lab, so they also can succeed. For me, and maybe it's a bit of a cliche, but my father was always a good role model to me, even though he's not a scientist. He is an academic, however, a professor like me and like my sister. His expertise is in Victorian poetry, though, and he's interested in history. And he's really congenial, has a great sense of laugh and a great sense of irony. He also has a strong sense of fairness and a healthy suspicion of power and great appreciation for tenure and a strong distaste for college football.
Gotcha.
Growing up at the dinner table, we were always listening to the shop talk of things that were going on in my parents' days at work. And so I learned to appreciate politics of academia and the process of publication and writing.
Maybe a little bit about Victorian poetry along the way.
Maybe a little bit, not too much. But I did grow up thinking that I might also go into the humanities, as that was really the emphasis in the family. And I was a pretty voracious reader for a period of time. And I think that taught me the value of good writing. And that's a lesson that paid off when I became a scientist. So writing and this appreciation for the importance of reading and critical thinking, they're all really important in science, particularly once you've directly left the bench yourself and you're leading a group. And publications, grants, reviews, these are the currency in which scientists work, and they all require excellent writing skills. So I think my father taught me a fair amount about the importance of good writing and thinking. And they also gave me a fair amount of latitude growing up. And I tried to do the same with my lab members. That is, teach them that writing and critical thinking are skills that are maybe not emphasized when you're training as a scientist, but are pretty important to success.
Certainly. And I think there's skills sometimes that are really hard to teach. There's no easy lesson plan for how to teach critical thinking. So do you have any tips for people who are maybe trying to learn these skills themselves or trying to teach them to other people in their labs or in their life?
Well, I spend a lot of time writing on papers together with my lab members. So we'll sit behind a computer together and spend, seems like an inordinate amount of time, maybe an hour per paragraph. But that allows us to hammer through difficulties and make sure that we all have a really good understanding of what we're saying, the message that we're trying to deliver. And so it takes a lot of time, but I think that's the only way to do it.
Sounds like a painful but effective process. So you mentioned that your father was an inspirational figure and that you maybe had an early interest in the humanities. Can you remember when was your first memory that you can point to of you becoming interested in science or maybe starting to consider this as a career?
I always liked being outside when I was a kid, and growing up in a household that was not so scientific, but academic, it maybe pointed me in the direction of being a professor. But I think that the choice of going into science was sort of a departure from the environment that I was raised in. In fact, it didn't really occur to me that I could be a scientist until I got to college in my freshman year and realized that humanities and the social science were not really my thing. I took a biology class and thought that maybe that might get me a job that was connected to the outdoors and nature, maybe something a little bit more concrete than the humanities, at least to me. Mm-hmm. It's perhaps a bit ironic that I ended up as a computational and molecular biologist. So we're studying things that we can't see, working mainly in a lab, and all our data is in sort of a virtual world. Right. So not outdoors, but we are working on plants.
Gotcha.
In any case, I kind of realized that I didn't want to go into medicine or medical research, as my passion was really with plants. And in high school, I'd worked in a restaurant for a number of years, so I felt a connection to food production. And in college, I was at a university that had a lot of research opportunities. And I took some classes with plant biologists on the faculty and got a summer job working in evolutionary biology, looking at inbreeding. And then one summer, I think it was after my sophomore year, I decided to get into agriculture a little bit more. And happened to read an article in the newspaper about an innovative organic vegetable farmer in central Missouri. This was about 20 minutes outside of a small town of 1,000 people, close to Lake of the Ozarks. Really a beautiful area, quite rural. And the job had a lot of hard work. So we were up at maybe 5:30 in the morning, working till noon, moving irrigation pipes, weeding and planting and harvesting. And then the afternoons we were washing, packing, seeding. Really tough work, but quite rewarding. We grew, I think, over 150 different types of plants that were mainly vegetables sold to high-end restaurants in the Kansas City restaurant market. And this made a big impression on me. It was really fascinating to see the diversity of the plants, the change of the growing season, and the tremendous effort that went into producing high-quality food. And at that point, I was sold on plants. And I thought maybe I would want to be a plant breeder to sort of focus on how that diversity is developed and utilized. And then fast forward a few years to graduate school, I moved to California and was working in a lab that did both breeding and molecular biology. And what I learned was that breeding required a certain patience that I didn't have. But molecular biology offered me puzzles to solve and maybe more immediate rewards and gratification and less statistics. But for those first few years as a graduate student, I was in California and enjoying the Sierra Nevada mountains and other outdoors activities. And I probably spent a lot more time hiking and rock climbing and skiing and rafting than a graduate student should.
Mm-hmm. It all sounds wonderful.
Yeah, it was. But it gave me really good appreciation for time outside of the lab and the type of thinking that stimulates. So that's something that I try to encourage in my lab members as well.
I think sometimes you do just have to step out of the lab, get some fresh air for a little while, and then you come up with some creative solutions or ideas.
Yeah, you need a good break.
So then you went on to do some interesting sounding postdoctoral fellowships, and one of them at Dupont Genomics was more of an industry postdoc. So can you tell us a little bit about that?
Sure. So I wanted to do a postdoc. Well, I wanted to try industry and see if that might be my career path. And I was fortunate in that my PhD advisor knew someone at Dupont, and they had an opening, and it was a pretty good fit with my skills. So I went and worked at Dupont for about a year and a half. So it wasn't there for that long as far as postdocs go, but it was really good experience. And I took away a couple of really important things. One of which was an exposure to a really novel and unusual method for DNA sequencing. And this method generated hundreds of thousands of short sequences from fragments of DNA. Today, we know this as next-gen sequencing. But at the time, I think they were the only group that was applying this in plants. The other thing I took away from Dupont was that I met my wife, who was also a postdoc there.
I love it.
So then I went back to California and did a second postdoc back in academia. And one of the things that I found was that the technology that I'd seen at Dupont wasn't being used in academics. This was in part because it was a pretty unknown, esoteric sort of technology, and it was also quite expensive. But I met a program officer from the National Science Foundation who managed a grant program who was funding the work that I was doing as a postdoc. And I described to her the method and the data that it produced, and I showed her my enthusiasm for it. And she was quite supportive and encouraged me to write a grant proposal, which I did, even though I was still a postdoc at the time. And that was kind of an eye-opening experience. The reviewers were pretty harsh. One of them suggested that maybe it was illegal for me to be a PI on a federal grant as a postdoc.
Really?
Which it wasn't. Another suggested that it was maybe poor training experience for me, which I don't think it was in the end. And another reviewer said that the technology offered no advantages over the technology of the time, which wasn't true also. Mm-hmm. Another comment was that there wouldn't be much of an impact.
Negative comments are raining down on you at this point.
Exactly. But at NSF, the program directors have some flexibility in their funding decisions. The reviewers only make recommendations to them. And I think she saw the promise in the work, and I got funding to try it out. And the technology and the company that I worked with was really the predecessor to today's next generation sequencing technologies. This was probably in 2002, and there was a quarter or two when my project was the main revenue source that sustained that company. But that company has gone on, and now it's over $20 billion. Illumina, which is one of the biggest players in sequencing.
Yeah.
And so I like to think, maybe a little immodestly, that early project laid the groundwork for this explosive growth in DNA sequencing that's driven genomics in the last decade.
Well, that is a fantastic story. I love this passion that you showed, this sort of innovativeness of starting to apply for those grants at the level of a postdoc. I know even as a faculty member, they're pretty intimidating. So very impressive. So now, can you tell us a little bit about how these two different postdoctoral experiences from the day-to-day life experience, how is the industry postdoc similar to or different from your academic one?
Well, in industry, the focus is on getting projects done and ultimately on a commercial application. I was fortunate in that the group that I was working in wasn't so focused on the immediate commercial applications. And at that time, they were really trying to get the sequence of the Meyers genome, which took another maybe 9 years to complete. But they were working on different aspects of DNA sequencing. And the nice thing about being in a company is that you could find experts who have skills in a really wide range of techniques and questions. And so you can get work done really quickly because the company works essentially as a large team. So if you have a problem, you can go over to another group or another division, particularly in a large company like Dupont, and tap into someone's expertise. And so as a postdoc, it's really great because your work goes very quickly. Budgets tend to be pretty high and funding is less of an issue.
It sounds very refreshing.
Yeah, it is. But you are kind of constrained in the questions that you can ask. Whereas in academia, you can ask whatever questions you want as long as you can get the resources to study them. So resources are more limited and you have to work harder to get the grants to answer those questions, but you have a bit more latitude and freedom in the questions that you're asking.
Gotcha. Well, wonderful to hear about these two experiences of yours and some of the fantastic things you've done along your career path. And I understand you are relatively new to the Danforth Plant Science Center. So can you tell us what brought you there and what's it like working there?
I just started last month, so I'm pretty new. I was at the University of Delaware for just over 13 years, and that was a pretty productive time in my career. Had some excellent colleagues, collaborators, lab members, and I started to look around and think about moving, and I spoke to some of my colleagues that had moved, and I think the message from all of them is that moving a lab is pretty disruptive. Mm-hmm. It takes time, maybe a couple months when you think about shutting down the experiments you're doing and then packaging everything up, and moving it and then unpackaging it and
ramping everything back up. Yep.
Yeah. But it's also pretty refreshing experience, healthy, shake things up, provide a new perspective, get new colleagues, think about new questions, think about some of their questions and how your work can impact their questions. And in terms of moving to the Danforth Plant Science Center, it's really a remarkable place. The infrastructure here is just world-class. Greenhouses, I'm looking out my window and I can see about 30 greenhouses lit up with thousands of plants in them. And so for a plant scientist, this is, I think, really the place to be these days.
Sounds like paradise.
yeah, a lot of growth going on. And particularly in this area of the country, Midwest, of course, is where a lot of agricultural products come from. And St. Louis is sort of a hub for ag biotech. And so I'm excited to be here and have colleagues both on the academic side at the University of Missouri, the industry side, and the nonprofit side here at the Danforth Center.
Gotcha. Well, wonderful to hear more about the Danforth Plant Science Center. And you said you're in the process now of ramping everything back up, getting your lab up and running. So can you tell us about one of these things that's going on in your lab or starting to take off in your lab that you are just the most excited about?
These days, I'm really excited about reproductive organs in plants, like flowers in particular. And I'm not a developmental biologist. I sort of got into the field that I'm in now, which is small RNA biology, through the technology side, as I described. I'm still learning quite a bit more. And of course, as scientists, we're always trying to push the limits. And so that requires learning a lot about new things all the time. But what's been exciting for me is that we've characterized some really unusual pathways for the production of RNA. And these are really enriched in anthers, particularly in the grasses like rice and maize. So anthers are the pollen-generating male reproductive organs in the flower. And they're interesting structures for a variety of reasons in that they produce innumerable pollen grains, and those are the gametes or products of meiosis. And what we noticed is that there are several rapid and extreme bursts of RNA production, small RNAs in particular. And the function of those bursts is still unknown, but we're learning that these are quite conserved all the way back to the origins of flowering plants about 200 million years ago. And so what's been fun about this also is that we're seeing some similarities between plant and animal reproductive development. And we think this might be a case of convergent evolution between the different kingdoms. That is, we don't think there's a common pathway that led to these bursts of RNA production during reproduction in both animals and plants. And the other exciting thing is that working in evolution has gotten us out to some of the great public gardens and plant collections so we can get samples from flowers of very diverse species. For example, when we were at Delaware, Longwood Gardens was just up the road from us. And so we went and worked with a taxonomist there who helped us collect flowers from their meadows and forests, and they have a really large conservatory. So we were collecting things like orchids and flowers from the giant Amazonian water lily.
Oh, wow.
Relatives of banana. And now being here in Missouri, there's Missouri Botanical Gardens that's in the city not too far from here. So we're looking forward to collecting there as well.
Fantastic. So can you give us an idea of really what makes a small RNA a small RNA? How big are they and how are they different from the other types of RNA?
Sure. Small RNAs are about 21 to 24 nucleotides in length, and there are a variety of different pathways that have evolved for the sole purpose of producing those small RNAs. And typically a messenger RNA, a more typical RNA that encodes a protein, might be anywhere from 500 base pairs to several kilobases in length. Mm-hmm. So these smaller RNAs are produced from a precursor that gets processed by one of these proteins, like a dicer protein that chops it down from a larger length, maybe 1,000 base pairs, to 21 nucleotides. And then they get loaded into other proteins called argonauts, and those argonauts use that small RNA to perform a sort of homology search and find the targets. And those targets then get sliced by the argonaut, and that causes this sort of dialing down of the RNA levels that I mentioned earlier.
Thank you so much for sharing more about the exciting research going on in your laboratory. And it sounds like you have some fun upcoming sampling projects going on at the Botanical Garden, so we'll look forward to hearing more about those. But I think so far we've talked about a lot of the great aspects of your career and haven't touched on as much some of the challenges and struggles. And I think these are very common in science, and it's important to show people all the struggles, all the failures that happen behind the scenes when most of the time what they see is the successes. So I want you to share with us today a story of your own failure or challenge or struggle and how you got through it.
Certainly. I guess the project that sticks in my mind is the one that we're most excited about. That is these small RNAs that we find in the anthers. And we had a really difficult time getting that published. Typically, the process of publication is quite fraught with challenges. You're trying to get your reviewers excited about a discovery that seems obviously exciting to you, the author. And so you submit your finished manuscript and you're sure that the reviewers and the editors are going to be leaping with joy over the contents.
Right. This is the coolest thing ever, of course. Yeah.
Yeah, exactly. Accept your manuscript with no revisions because you've spent so much time polishing it, like that 1 hour per paragraph that I mentioned. And then sent back to you, declined, maybe even stopped short of the review process by an editor who fails to see the merit of your work. And that was exactly our experience with this paper about the anther small RNAs. We finally published it last April, and we were pretty excited about it. It seemed like a really novel, really insightful, complete story to us. But before it found a home in the journal, even before it went out for external review, we spent almost a year submitting it to different top journals, revising it, adding more data, polishing it. It was pretty demoralizing for us because some of the editorial comments made us feel a bit like plant research doesn't always get the same level of respect as biomedical research, which is not an uncommon sentiment.
Sure.
Anyways, I'm really proud of the paper, and we got it published in Proceedings of the National Academy of Sciences, so quite a good journal where it's visible to scientists outside of plant biology. But it was that year that we spent polishing, revising, submitting, getting rejected. It was a tough time trying to keep the students and postdoc who were the first authors on the paper, trying to keep their chin up, as it were, because we were so excited about the work. We just couldn't see how the editors didn't appreciate what we were describing. So maybe it was some fault of our own in how we described it, but that's part of science.
Well, and I'm sure this is resonating with a lot of our listeners out there. I know firsthand that publishing experience can be so frustrating sometimes. So how did you cope, or how did you respond when you got a rejection back?
Well, I guess it was a matter of sitting back and regrouping and thinking about what we were going to do next. In some cases, we did argue with the editors because for those 8 or 9 journals we sent it to, in most cases, they weren't sending out the paper for review. So we weren't even getting comments back from 2 or 3 reviewers. It was just the editor that was making a decision. We tried arguing back and forth, and that wasn't very successful either. So it was kind of a unique experience for me because while most papers take a submission or 2 to find a right home, a right journal, that level of repetition of rejection was quite remarkable.
I think that's where that persistence comes into play. You have to keep trying. You have to keep submitting it, particularly if you are that passionate about the work you're doing. So I'm glad to hear that you were able to turn this challenge into a success. And I love celebrating all of these successes in science in their many different forms. So I want you to share with us today another successful story of yours.
I guess I would point back to the same failure and say that the success was ultimately getting it published, because now the work is out there. We've described this, what we call a spatiotemporal map of how these small RNAs are produced in the anthers. So we put together a lot of data that described when these small RNAs are being produced, and we were able to use a variety of different mutants. So these are male sterile mutants of Meyers that my collaborator, Ginny Walbott, who's at Stanford University, she's been working on these for a number of years. And I think it was just an elegant study using the mutants and then coupling that with some microscopy to demonstrate the locations where these are produced. And so in the end, even though it was a series of failures, the outcome was this beautiful publication that we're quite proud of. So we're building on those results now, and that's sending us out to the gardens, as I described earlier.
Well, that is awesome. Congratulations. And now after this long, drawn-out process, this sort of back and forth, how did you finally celebrate this success?
Well, there was certainly some meals and beer that that went with it. It was a collaborative project between my lab that was at Delaware and Ginny's lab at Stanford. So we were on different coasts. And we find that we do a lot of collaborative work with scientists all over the world. And so it's a little challenging to have the sort of face-to-face celebrations with the immediacy of getting your paper accepted. We often see our collaborators at meetings, conferences, maybe once a year or maybe twice a year, that sort of thing. So conferences are a really nice way to bond over these types of successes.
Absolutely. And I know we talked in the beginning a little bit about your love of travel, and I think conferences are another great way to travel in science. And I love talking about all these fantastic places that people get to go for science, whether it's for fieldwork or conferences or what have you. So can you share with us today, Blake, if you had to pick just one favorite place that you've traveled to for science, where would it be and why?
I'd have to say New Zealand. It's been maybe over a decade since I was there, but I went there for a conference. My wife went with me, and we took about 2 weeks of vacation and traveled around both islands, the North Island and the South Island. And the geography there is just incredible. It's really varied over a very small amount of geographical space with incredible coasts and forests and glaciers, tree ferns, volcanic regions, spectacular diversity, really unique species. And it's in a country that closely controls their development, or seems to for a foreigner, and doesn't have a high population pressure. It made me think that it was perhaps like what California must have been maybe 80 years ago or so, before the cities had grown and highways had sprawled across parts of it. But I also spent a lot of time in California in the mountains. There are a lot of volcanoes in California and volcanic activity. And I saw that also in New Zealand. It's just a really spectacular, amazing place.
Certainly. I've been to New Zealand myself, and I was struck by just how green and beautiful everything is. So was there a favorite thing that you did while you were there? Or maybe a place that you were able to hike around?
We went to, I think it's called Rotorua, which is a volcanic area that's like a large volcanic depression that has a lake and
sulfur springs that smell wonderful.
Exactly. Just really rich colors from all of the minerals, some steam vents blowing out, and you get up high and the air is brisk and refreshing. It was remarkable.
Fantastic. Well, it sounds like a wonderful experience there in New Zealand. And I must say, listeners, if you're able to travel to New Zealand, it is a fantastic place, and we both recommend Rotorua. So, and now we've chatted a little bit about your life outside of science and some of the things you like to do. And you mentioned that your father instilled in you perhaps a love of reading and literature, and at least the importance of those things. So I want to chat with you a moment about books. I love getting book recommendations from just about everybody that I talk to. So Blake, can you recommend to me a book that you've read and really enjoyed? And this can be a science book, a non-science book, whatever you like.
Yeah, I have a couple of recommendations. Both of them are historical biographies. One of them is called George Beadle, An Uncommon Farmer. It's by Paul Berg and Maxine Singer. So George Beadle was a geneticist, and he was born and raised on a farm in Nebraska. And he went on to earn a PhD working on maize genetics, or corn genetics. And over his career, he worked on corn, he worked on Drosophila, so fruit flies, worked on a fungus, Neurospora, the bread mold, which is a great model for genetics. He ultimately won the Nobel Prize with Edward Tatum for testing the hypothesis that one gene encodes one enzyme. And what was neat about him, he was just a very delightful, humble individual, won a lot of accolades, and was a scientific and academic leader. And there's some really classic stories in the book about things like the time he was mistaken for a gardener when he was actually the president of the University of Chicago.
Oh, wow.
I think he was out in front of the president's house wearing his overalls and tending to the plants. Mm-hmm. So he looked like a gardener. It was just his passion for plants that he wanted to get out and be outside. Another book that I really enjoyed was called Tuxedo Park by Janet Conant. And it's about the life of Alfred Lee Loomis, who was a wealthy philanthropist in the 1920s and 30s and 40s. And he used his wealth to support a number of scientists and projects. His support led to a number of both really basic and really practical discoveries, things like radar, which was incredibly important during World War II. And he was just a very insightful person and used his great wealth for the benefit of many people, mainly because he enjoyed science and had a great respect for scientists. So both of those books, both biographies, as I said, are really enjoyable.
Well, fantastic recommendations, Blake. I'm curious, do you often find yourself reaching for these more biographical books or nonfiction type of books?
I do find myself reaching for the biographical books. I used to read a lot of fiction, but maybe my attention span is shorter. And knowing more scientists now, it's interesting to relate some of the personalities that you read in the books to people that you run into through work.
Certainly. And I think these personalities, there is no shortage of them in science. There are so many fantastic, wonderful, funny people that I've met as a scientist. And I love talking about the stories of kind of what happens when you put all these people together. So I think there are a lot of fun traditions that develop, or just hilarious memories that you share together. So can you tell us one of your own stories of the human side of science?
Well, I'm probably not the best person to ask about quirky experiences. I think I work a bit too much. And as they say, all work and no play makes Jack a dull boy.
Yeah.
But I try and maintain a pretty relaxed and supportive, fun atmosphere in the lab. So I like to laugh. I like to joke a lot with my lab members and colleagues. We have a pretty culturally diverse lab, people from all over the world. And it's fun to share what's unique to our different cultures and maybe try to teach people in the lab a few quirks about English language and just funny expressions and things like that. So while I enjoy a good joke, it hasn't led to any quirky lab traditions quite yet. But there's still time, so I think I'll work on that.
So can you tell us about maybe one of these cultural or traditional experiences that you've shared between yourself and other people in the laboratory?
Well, when working on the papers, for example, Like one expression is to throw someone a bone. And so I remember working with one of my Chinese students and telling them that we need to throw a bone to the reviewers. And I think that the expression, it was not immediately obvious what it meant.
Right. You take these for granted too. Yeah.
Right. So then we ended up, and that became kind of a common tagline when working on the revisions for that famous manuscript, is that we were constantly trying to throw the reviewers a bone when they were asking for some really heinous experiment that was going to take us 2 years to do, where they had a good point, but we didn't want to spend the 2 years to do the experiment. So we would try to give them some bit of data that maybe partially addressed their concerns.
Right. And then gradually guide the conversation back to that is out of scope of.
Right.
Well, that sounds fantastic. And it's great to hear that you've been able to put together such a diverse laboratory and bring people in from all over the world to work together. So wonderful to hear more about the people in your laboratory now. And we've talked a little bit about some of the research that you're doing now and some of the things that you've done previously. But I want you to dream big with us today, Blake. If we gave you everything right now, money, staff, technology, feasibility, not a problem, what would you want to tackle?
Wow, what an opportunity. So I would probably point back to one of Charles Darwin's questions. And in fact, this is what later got labeled his abominable mystery. And so what he had noticed was that flowering plants are incredibly diverse. There are hundreds of thousands of species, and they comprise maybe 90% of all living plant species today. Yet they only emerged in the last 200 million years, so just a small slice of the history of the Earth. And yet there was this veritable explosion of diversity in the last 100 million years that led them to dominate the planet. And what was pretty mysterious to Darwin was that plant evolution was generally pretty slow and steady for hundreds of millions of years. And then in these last 100 million years during the Cretaceous period, flowering plants just exploded in diversity. So why did it happen? Why was it so fast? And this was Darwin's mystery, his abominable mystery. And maybe with today's technologies like genome sequencing and analysis, we could actually answer that. We'd need to sequence lots of plant genomes, representatives of all the major branches of flowering plants, of course, some of the gymnosperms, that is the seed plants that predated the flowering plants. And so I think with all those genome sequences, I'd be able to answer a few of my own little esoteric questions about how microRNAs evolved and diversified and the role in the evolution of plants. So while this isn't exactly Darwin's abominable mystery, if we had all those genomes sequenced and enough primitive angiosperms and their earlier ancestors, pine trees, for example, I think we could work out pretty effectively how flowering plants evolved, how flowers themselves evolved, how the developmental patterns evolved, what sort of innovations were required, and how they've diversified over the last 200 million years or so. I think that would be really exciting.
Very cool. I like it. So what do you think is the main limiter here? Is it just sort of time and manpower, or?
Well, given the cost of sequencing, which has come down tremendously in the last 10 years, it's maybe a matter of time before all of these, or at least many representative genomes, are sequenced. Even today, it seems like it's maybe every 2 weeks that a new plant genome gets sequenced. That's just a torrent of genomes compared to going back maybe over a decade ago. The first plant genome was sequenced in 2001, and it was, I think, another 3 years before the second plant genome was sequenced. And now we're at a pace of about one every 2 weeks. So if you fast forward 5 years, maybe we'll be getting a new genome every couple of days. It is just a matter of time, and we will be able to answer these questions. But I'd like to do it sooner if I've got all the resources at my disposal.
Definitely. Well, that is so cool. And I love the term abominable mystery. So if you could go ahead and solve that, that would be fantastic. Well, Blake, it's been great to dream big with you here today. And I know we've talked about a lot of things in our conversation, and I love to leave our listeners with advice, something that they can do in their lives now to maybe help them along their own journeys. So can you think back to all the advice that you've received and share with us one piece of advice that you think is just the best that you've received?
Definitely. I remember graduate school was a really tough time, and I think it's a tough time for a lot of people. And I actually tried to drop out of graduate school about halfway through. And my graduate advisor pointed out that my career prospects in science were probably better than my plans, which were to become a ski instructor.
Ooh.
And so I think he was right, although there are definitely occasionally days when being a ski instructor
sounds pretty good.
Yeah. So his advice was that science is a pretty good career, and I should stick with it, even if it gets tough, which it often does. So I guess I would encourage the listeners to stay with it, even though it is a tough career. It's quite rewarding.
Well, excellent advice. Listeners, definitely stick with it if you are going through one of these tough times. Now, Blake, is there any other piece of advice or a last note of inspiration that you want to leave our listeners with today?
Certainly. I think that plants are really critical to the survival of humans, really. They're a source of our food, our fiber, and our fuel. They formed our atmosphere. They're also really beautiful organisms and eminently practical in terms of agriculture. And there are a lot of scientific mysteries that have yet to be explored, and answers to some of those mysteries that may seem like sort of minutia or overly esoteric, like maybe studying microRNAs. I think that some of them will provide practical, real-world solutions to problems that we face. Maybe this isn't so often appreciated by politicians, and as a country, the United States hasn't been investing enough in plant science to make the sorts of discoveries that we need, both for commercial applications and to stay competitive in the global marketplace for scientific talent and ideas. I would encourage people to let their politicians know about this, and also encourage students, postdocs, to study plants, because I think there's a lot that we have yet to learn.
Certainly, and definitely take a moment to appreciate the plants in your life. Like you said, there are food, fuel, fiber. They provide so many resources that we use on a daily basis that we might be taking for granted. So I'm glad you brought that up, Blake. Wonderful to chat with you. Can you tell our listeners now how they can reach out if they want to learn more about you and the work you do?
Certainly, I would say that email is pretty good. I can't always promise the snappiest of responses, as it seems like I'm always behind. I'm listed on the website of the Donald Danforth Plant Science Center and at the University of Missouri in the Division of Plant Sciences, along with my contact information. The Danforth Center is on social media, so you can chat with them on Twitter at Danforth Center, and the website is www.danforthcenter.org.
Perfect. Well, listeners, definitely get in touch. Check out the website. Blake, thank you so much for joining us as a guest today.
It's really my pleasure. Thank you for the opportunity.
Well, it was great to chat with you. Listeners, great to have you here as well. We'll see you next time on another episode of People Behind the Science. Your voyage to explore the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guest favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to chatting with you next time on People Behind the Science.